Welcome back everyone to the lecture about applied bioimage analysis. This is a summary of the whole semester. So first of all, I would like to thank some people. So I'm putting this acknowledgement slide at the very beginning because I think it is actually quite important to tell you that it's not just me sitting here talking about some slides. There's actually a lot of people behind who prepare slides, who organize courses, who share material, share software, stuff like that. So I cannot go through all of them. And I'm sure I missed some here on this slide. But I wanted to express some thank you to all the people who shared material with me, who taught me everything I know so that I can give this further, and also on a European level in the new bias community, organize teaching in a larger scale so that we can share knowledge with each other and are not sitting, every one of us, in our kitchen trying to figure out things on our own. Furthermore, there's also the software developers and also the people here who shared material with me from all these different toolboxes. So they keep the machinery running. So it's them who allow us to use software for free, which allows us to process our data. So without the names here on this slide, we would likely sit there with C, C++ and write our code ourselves and be much less efficient than we are thanks to open source software and to people contributing to open source software. And with that, I will give you a short rush through this semester. Image analysis is a part of the experiment. So when you again think about the observation you have, we see something that's interesting, Drosophila, fruit fly here in this case, and we decide to set up an experiment, do some microscopy, and look at development of a Drosophila embryo, for example, in more detail. And then we do some image processing in order to convert our image coming out of the microscope into an image. For example, here you see a label map, a parametric image, an abstract representation of the scene we have observed with the microscope. And then we do image analysis in order to turn this abstract representation image into non-image data. For example, in a box plot describing how large cells are in different regions or if different animals. Then we do some modeling. We try to represent what we did, what we measured in an abstract model, and then we can draw conclusions here and figure out how the thing works. And in order to prove that everything is indeed as we modeled it, we have to do a new experiment. And especially for young scientists, it's often important to mention that again and again, that this circle is an absolutely normal thing. The fact that we have to go back to the microscope and take another image is not does not tell us that we failed in doing it in the first moment. No, it's the normal way of doing this. We go through the circle over and over, learn and learn more, and after some cycles, we have a much better understanding compared to the initial observation we had. So going through the circle, analyzing things again over and over, taking new images, making new models which are different from the models before, is the normal way how science works in this context. Just to iterate on that, so when you look at this typical image processing workflow we were doing a couple of times in this semester, you start at acquisition, you go through a denoising step, you do background subtraction, you do segmentation. Maybe after segmentation you do a correction of the binary images before you do the labeling. And then by the end you do feature extraction and descriptive statistics and stuff like that. So in each of these steps, things can go wrong. And that's why it always may make sense to go back. That's what we in ImageJ did all the time. By you open an image, you apply a filter, you realize, nah, that was not the right thing, so try a different filter. So it's like, apparently this is failing, but in fact, this is reiterating on the same workflow and continuing to improve the workflow in order to find an optimal solution, in order to find a good way of actually quantifying what we originally had in the image. So again, has nothing to do with failing and being bad in image analysis. This is the normal way of doing it. And the more experience you gain in processing images, the easier this back and forth will become for you. Then we discussed about some very general terms of how image analysis or how science is supposed to be. So we want to be quantitative. We want to make numbers out of our images and not terms like many or not so many. We want to have an actual number. Like in this image, there's 3 mandarins, not many or something like that. And we want to be objective. So we want to have a computer doing a measurement, and ideally a fully automatic measurement of a scene or of objects in an image without a human interacting with it, without manual annotating or stuff like that, to be as objective as possible, then the whole workflow must be reliable. I will also tell something about the differentiation between reliability and validity. A student was asking for that. So we want to have trust into what our workflow is doing. So we have to kind of prove to ourselves that what our automated workflow, our software is doing here makes sense. Then we want to be reproducible. So we want to document our workflow in a way that somebody else can do it and would draw similar conclusions as we do. And furthermore, repeatability is a property of the experiment. Yeah? So if we sit in front of our computer and we execute a program twice, this is 100% repeatable. Ideally, also there is no random numbers in our algorithm, and that's why the results are still the same. But if you perform an experiment at the microscope, where, for example, light destroys the sample while acquiring an image of it, then this is not repeatable. So these kind of terms are important in order to classify workflows and to have some kind of ideal conditions under which you can actually draw good conclusions from data from acquisitions. There was a student asking via email for basically for the difference between reliability and validity. And actually, so it basically means the same with an additional aspect I would like to introduce a little bit more in detail in order to clarify understanding here. If you think about an algorithm and you want to kind of show that this algorithm is doing the right thing, it delivers the right results, you can, in some cases, mathematical algorithms, you can verify the algorithm, you can prove that the results are correct. If you, for example, have a mathematical notation of the algorithm, so then you can make a mathematical, a logical proof under these conditions. These are the results, and the results are correct because of an argument list you can provide. So this is in pure in mathematics, in logic, you can do this. So this is a technical approach, let's say. In computer vision and also in biology, you sometimes cannot do that because we are relying on a fuzzy biological thing which we have to manipulate manually in order to put it in a microscope, and we can then measure the size of it. So we do not know the size of this thing, and that's why we cannot prove in a mathematical sense that the size we measure with our image analysis workflow and with the microscope is really related to the actual size, because we don't know the actual size. But we can make samples, or we can take samples which are produced by companies, for example, fluorescent beads of given size, and perform additional experiments in order to validate our strategy. So if we measure with our microscope and our image analysis workflow objects of given size, and can then show that the size which we knew before and the size we measured are related, are strongly correlated, and then we can validate our algorithm, and we can tell something about the validity of the algorithm. So, and then there is this term of reliability. I also sometimes call it trustworthy, because if we take an algorithm which was maybe validated on a particular data set, and where a mathematician maybe proved initially that theoretically under certain conditions this algorithm does the right thing, we still do not know if it's reliable on our data, because our data may be different from what this thing was thought for, and we may also have between different data sets fluctuations no mathematician was ever thinking of when developing this algorithm. And that's why I interpret the term of reliability or trustworthiness as our subjective, it's a subjective thing, our subjective trust or feeling with this algorithm applied to our data. And so the student who asked for that also argued that the definition of reliability also has something to do with reproducibility and repeatability. Of course, if my algorithm is not repeatable, yeah, so if I do not get the same measurement out when applying the algorithm twice, then I would never trust it. So that's absolutely right. It's absolutely correct. So how can we increase reliability or trustworthiness of an algorithm? And there I would say it has something to do with visualization. So you see here a video, we were processing in lecture number 4, and you see a plot resulting from this video. And when I look at the video and when I look at the plot, my trust in this strategy is not high because I do not see the direct relationship between my data set and this plot. This plot can be right, this plot can be wrong, and I don't know. So I'm sitting in front of my computer and I develop workflows all day. And when I develop a workflow like that, I'm not happy because I don't trust it. I don't think that this is reliable yet. And there visualization of the results play a big role, and this may also lead you to do different measurements. So we changed in lecture 3, lecture 4, we changed our strategy from measuring the mean circularity, where we don't know if this means something, to the number of round cells in this image. And the number of round cells in this image, in this video scene, we can actually at every time point stop the video and count and correct for that and gain trust. And this is what I mean with reliability. I developed a workflow from a theoretical point. The algorithm does the right thing. It was proven under different conditions that this algorithm is good. But when I apply it to my data, I have to do some additional effort in order to make it reliable, in order to gain trust in what I'm doing here. And visualization is, from my point of view, the key in order to doing this. When we are talking about visualization, we also have to talk about lookup tables. So there are lookup tables which are better suited for visualizing my data, and there are lookup tables which are not so well suited to visualize my data. And if I, for example, want to show an edge between a dark region and a bright region, then I may take a lookup table which has many colors instead of gray and black and white. Yeah, so and always keep in mind when you apply lookup tables to your data set, they do not change your dataset, they just change how the dataset is visualized on screen. Furthermore, also for inspecting image data, you can look at image data, you can change lookup tables, but you may also want to do a summary of your dataset. So you see here, for example, histogram. So what is a histogram in context of pixels? And you see here that we basically only have grayish pixel values in this image, and maybe some black, and maybe a peak in a brighter gray, and that's what we actually also can see in the image. And I change the configuration of the microscope, the contrast becomes higher. Higher contrast means a wider distribution of gray values in this image, and it also shows here that we have now this additional peak here by the right has gone, and we distributed these pixel intensities over more different gray values, and that's why we can suddenly see nuclei here in this original image. So the histogram tells us a bit about image quality, so differentiability of different objects. You can see that in the histogram. Then a reminder of a terminology thing. Pixel size is not resolution, even though they are related to each other. So if I have very large pixels, that would mean that I may not be able to resolve small objects. But having very small pixels may not allow me to differentiate these small objects if the resolution of the microscope is not enough. So these two images here, I zoomed in. These two images have the same pixel size, but the resolution of the dataset behind is different. That's why when I draw here a line and plot the profile along the line in this image, I can differentiate these two green objects here in this image, and in this image I cannot. Furthermore, we were talking about pixels and voxels, the picture elements and the volume elements, and typically in three-dimensional imaging microscopy, voxels are not isotropic, so that's why you see in different visualizations of different planes. So you see here an XY plane, you see a typical image with pixels, some noise and some background. And when you look in the YZ plane, for example, you may realize that you have kind of these rectangular shaped pixel structures. This is a typical image of anisotropic voxel images. Then the Nyquist-Shannon sampling theorem, dependent on the resolution of your system and dependent on how many pixels you take per micron, you may be able to see an object or you may not be able, or you may see different objects glued to each other just because your resolution is not enough. So keep this rule of thumb in mind. If you want to acquire objects which are something like 3 microns large, then you should at least acquire a pixel every micron, if possible, if your microscope allows to do so. Furthermore, we were talking about multidimensional image processing, image data. There are different things which should be taken into account when talking about dimensions. So space and time are pretty obvious. In microscopy, we also think a lot about channels because we acquire different aspects in the same space and then save them as channels, as for example, color channels corresponding to the wavelength, which allowed us to acquire a certain image. But we also have to think about we have actually many samples, which is another dimensionality. We may also have images of different species, of different experimental conditions, mutant controls, right? So stuff like that. And last but not least, when we process the images from original images to some quantitative information, then we have additional images expressing the quantitative information, which makes it even harder to analyze this whole bunch of dimensions of image data. So how do you deal with that? So you should, if you are interested in a particular region of your dataset, then you should crop out this particular region and not process the whole image, because that makes things faster. And making things faster actually means making things doable, feasible. And what you can do in space, you can also do in time. So if you have a long video sequence where in this particular moment something is happening, then crop out this particular moment and only analyze these frames in time. So cropping in space, cropping in time. You can change the spatial resolution. You can change the voxel size of a dataset by downscaling it. So analyzing less pixels where your objects are still visible allows you again to do it faster and may allow you to do it actually. You can do the same thing in time. You can drop every second frame if the thing which is moving or the process which is happening is not too fast. Furthermore, take projections into account. If you want to see how cells are touching or dividing, if you can see this in 2 dimensions, why analyzing a 3-dimensional dataset might not be necessary at all. So do maximum projections. You can also do chimeographs, which is a projection in time, to have an easier 2-dimensional view on your data. Before you look at a 4-dimensional dataset in the big data viewer, why not making a chimeograph, which shows you everything that's happening on one screen at one time? And the more you go towards big data, you want to process your data in tiles and you want to resave your data set in pyramidal file formats in order to make actually the analysis possible. And there's more and more tools appearing these days allowing you this kind of image processing. So keep an eye on what's happening. Furthermore, when we think about tiling brings additional challenges. So connected components analysis, for example, is an algorithm which is not very easy to do in tiled images because actually relationships between these tiles have to be taken into account. It's not that there are no algorithms for that, but the number of tools which allow you to do these things on tiles like connected components is actually limited. I expect that to change in the next years, but if you discuss with a developer while segmenting objects in 12 terabyte large datasets is not so easy. This is the reason, because some algorithms are actually pretty hard to tile, to process in tiles. Furthermore, when we talk about tiling, we also should talk about graphics processing units, because when you think about graphics processing units, they have more compute units, so they can do more things in the same time as a central processing unit. Also because these graphics processing compute cores are optimized for image processing, for vectors, for matrices, for stuff like that. That means processing on a graphics card is actually always kind of tiled, or you have to kind of be able to divide your problem into things you can compute on compute units on a graphics card. And if you think about the general differences between CPUs and GPUs, think about graphics cards have more compute units, that's why they can do more things at the same time. And second of all, usually graphics cards have faster memory, which allows us to read and write images faster than on a CPU, which is beneficial, especially when you think about processing big data. And that brings us to the subsection filters. And the most important take-home message when talking about filters or image filtering is filters don't do magic. It's just math. So why do we do image filtering? So let's assume this is our original image, and the human observer can easily see that there are something like 4 cells in this image. However, when we apply some thresholding to this image, the computer may see thousands of objects. So we have to pre-process this image, for example, by blurring, and then threshold it, and then the computer is able to differentiate these 4 objects in an easier way. This is why we apply filters to images. And when you think about how are filters applied, so applying a filter to an image is basically moving a region of interest or a window of some pixels, moving it over our original image and measuring, for example, the intensity of these 9 pixels, multiply these 9 pixels with these 9 factors over here, and then taking the result of that and writing it in a new image. The process we are doing here is called convolution. So we convolve the original image with a kernel matrix of that kind, where every entry here in this matrix corresponds to a certain neighbor pixel position and the weight, the factor, which has to be multiplied to this pixel in order to compute the new intensity in the new image. So convolution, we are convolving an image with a kernel matrix here in this case. And we call this a linear filter because there is a linear relationship between the original image and the resulting image. And this linear relationship, these factors, are written here in this matrix. If you cannot write down a filter, so this is a mean filter, if you cannot write down a filter as such a matrix, then you have a non-linear filter in front of you. Furthermore, if you want to figure out what a filter is doing, of course, you can read about the filter, you can watch a YouTube video where somebody explains it, but I would actually recommend the experimental strategy. So take an image with just 1 byte pixel. So you can get that by drawing a super small rectangle of size of 1 and adding an intensity of 255, for example, to a 8-bit image, and then applying a filter to it and looking at the result. For example, if you apply a Gaussian blur, think about the Gaussian distribution we saw later in statistics. We're entering, here we are not measuring the sigma, like in statistics, here we are entering the sigma in order to do something to the image that afterwards our clean pixel, which is kind of the ideal condition, the mean value of our population, to turn it into a distribution in this 2-dimensional space, where we can see that here was our original pixel and the values go down according to a Gaussian distribution, according to a normal distribution. This is what the Gaussian blur does. And actually what we see here, as we had just 1 single pixel set to something over here, we basically see here the filter matrix which is applied to this image. So obviously we can express this filter as a filter matrix, as a 2-dimensional table of numbers. And that means Gaussian blur is a linear filter. And furthermore, there was a question of what's the difference between noise removal and background removal. If you think about these 2 things, there's a very nice example of a filter which does both things at a time. That's the difference of Gaussian. Maybe you remember this one. So we see our original image. There's some noise present. So we see fluctuations of intensities everywhere. So it's not like there's just white and black. There's many different shades of gray. And we can also see that they are locally a lot changing. So this is what we usually refer to as noise. In order to remove noise, we apply a filter, usually with a rather small radius, in order to get the intensities more equal to each other in neighboring pixels. So this is noise removal. Furthermore, if we take a much larger sigma and subtract it from the original image, so this is from the original image or from the noise removed image, then we see only the objects are bright and the background is basically even black. This is background removal. So when you look here in the original image, there were some gray regions between these nuclei, which are now gone in the background removed image. So removing background is removing something big in the image, some regions which are brighter compared to other regions, making them all similarly bright. That's background removal. And on a very local scale, in small regions, removing these fluctuations of pixel intensities, noise, that's noise removal. So that's the difference between background removal and noise removal. Some student asked for how the Laplace filter works. So let's look again at a normal distribution, basically expressed here in this plot. And we now look at the change of the signal from the left to the right in every point. So it's basically the change of the signal is going up. So it's not linearly changing the same way from every point to every point. No, the change is going up. So the first derivative is increasing until, for example, you see here at -1 approximately. So you see basically here there's then a shift. So the change is going down again. So that's flattening the curve. And you see this in the first derivative by the value going down again, and it's still going down at this point until it returns again and increases again. So this is what you see here. This is the first derivative. And then we basically do the same again on this curve to determine the second derivative. And when you look at this one from the side, it looks like a Mexican hat. That's why the Laplace filter is called a Mexican hat filter. And this is all. We now looked at one-dimensional Gaussian distribution, a one-dimensional image, a one-dimensional signal. If we extend this one dimension in a two-dimensional image, we have to basically do it in two directions. So this is the first direction. This is my original image. I have just a white pixel here. Now I apply a one-dimensional Gaussian blur to it, so I get this result out. You see here in the center, it's white, it's high intensity, just like over here, and to the side it's going down. And this is not linear, this is a normal distribution, so we are looking here at the Gaussian curve. Now I take the first derivative of that. So I start at gray, it becomes bright, so it's becoming higher, then we go down again until it becomes black. This is happening here. And then we go back to gray. This is basically what the first derivative is. And the second derivative is then going up a bit from gray to white. So that's why we see white here, going to black, 0, no, negative actually here at this point, going back to bright. And if we now turn this into a two-dimensional thing, so we do it not just in X, we also do it in white. We see again from our original pixel was a single pixel white. We see here the Gaussian blur of it, and we see here the first derivative of it, and we see here the second derivative of it. Just imagine you have this Mexican hat in front of you and you're looking into it. It's dark inside. The closer you look to the rim of the hat, it's white outside. So that's why we are talking about the Mexican hat filter. This is basically the Laplace operator applied to images. And a very simple version, when you look at that, so we have here black negative values and here white positive values. You can also turn this around and just multiply it with -1. Then you have a filter kernel which is positive on the inside and around is negative. So this is a very simple version, a simplified version of the Laplace filter. And we see here, this is my original image. I convolve it with the Laplace filter and I get this image out, which is in this particular case not very helpful because it's an edge detection filter, and edge detection in a noisy image shows us a very noisy result. So that's why we have to do we have to apply some Gaussian blur first to this image and then do the edge detection basically to get the Laplacian of Gaussian out. And this is also some kind of background subtraction, some kind of edge enhancement. It's used in a couple of tools like TrackMate, for example, in order to get these objects easier detected. And for example, to do some spot detection and then afterwards tracking. Furthermore, it's maybe not of a daily practical use. My professors were infecting me with the idea some 10 years ago, and I would like to infect you with the idea. Pixels do not have to be squares. Pixels can have any shape, and you can apply filters like a mean filter or a median filter also to an image where not every pixel has 4 neighbors. Or 8 neighbors directly connected. It can also be 7, 5, 6, depending on how they are arranged to each other. So this is just a reminder of this from a theoretical point of view exists. You can also apply it in, for example, CLIJ And the idea is here to use this as an abstract representation of cells. We are working a lot with images representing cells. These cells are communicating with each other, they are exchanging signals with each other, and chemical signals, for example. And image processing in this particular way may allow us to understand better what is happening here on a cellular level and not on a pixel level. That brings us to image segmentation. So far we were applying filters in order to get a better image segmentation or get an easier situation for image segmentation afterwards. And if you think about image segmentation, so there is a couple of terms which are used here in this context. So I often refer to a binarization of the image as the segmentation step, and afterwards, if I want to label all these objects with different numbers, that's the labeling step. And there are different approaches like thresholding, machine learning. I showed you a bit more in detail, but there is also alternatives to that, which are then used in more sophisticated scenarios. If you think about thresholding basically means you take your range of intensities which exist in the image, and you say all intensities above this particular threshold belong to my object, and the intensities below do not. You can take the original image and threshold it. You can take a blurred image and threshold it. And when you think about what machine learning is doing, it actually applies thresholds to a couple of images at a time in order to figure out what are objects and what are background. And when you use thresholding, or when you use any kind of segmentation algorithm to your data, keep in mind that you want to apply the same strategy to all of your images. You don't want to change based on your human observation, perspective, subjective impression. You do not want to change the workflow which processes an image from image to image. You want to process them all in the same way in order to be objective. You should check your algorithm with a couple of images and figure out if your algorithm works on all of them theoretically, on a whole variety of images and not just on one image, in order to gain reliability here, in order to learn how to trust your workflow. And you should document what you are doing to your images. The simplest way of how to do this is you take the image.j macro you recorded while processing your images, and you put it in the supplement of your publication. That's the easiest way of documenting a workflow, and it allows you that others can reproduce what you are doing. Furthermore, there are some tricks. So if you realize that your thresholding is not working well, so for example, we see here that's our original image, we want to segment the cell, and when we apply some thresholding algorithm, it looks like that. And we are trying a couple of thresholding algorithms, and they all did something like this, so it didn't work. Then do some blurring, really like blurring too much until the actual structure inside your cell is gone, because you're not interested in the structures inside the cell, right? You're interested in the cell, and then do thresholding. But please also keep in mind that if you want to do some quantitative analysis, for example, of signal intensities in your cell, then please do these measurements in the original image and not in the blurred image. And furthermore, don't forget all the steps you can do between segmentation and labeling. So after you did some binarization, some segmentation, some thresholding of your image, you may still see some issues in your image, in your binary image now. So for example, here there's a black hole inside this embryo. It was originally the embryo data set. So in order to get rid of this, you want to, for example, apply the operation fill holes. Furthermore, you can use binary erosion dilation in order to improve the segmentation result before analyzing it. Also keep in mind the watershed algorithm, which allows you to cut here these touching cells into individual objects. And you may remember that there is a quite long list of different combinations of these filters. Maybe you want to open image.j, make a simple binary image, for example, out of blobs.gif, and apply some binary filters to it in order to recall what they were doing. So it's like there's dilation, erosion, opening, closing, fill holes, watershed. So there's a couple of those I showed you. Maybe you want to try them again and figure out what they are good for, and how they can be used in order to improve a segmentation result before you go further to connected components analysis. So there was a question by a student asking what's the difference between connected components labeling and connected components analysis. basically, it's just two terms used basically for the same thing. If you want to, so if you think very carefully about it, connected components analysis is finding out which pixels or which objects in the image are touching. And if you afterwards label these pixels according to which belong together, which are connected, then this is connected components labeling. But it is actually the same process which delivers finally a result of that kind. So we started a binary image shown here on the left, and we mark the first pixel, the first white pixel in this image we find, we mark with a number 1. So we are labeling this pixel with the intensity 1. And then we analyze which other pixels are connecting. This is connected components analysis. And we figure out, for example, that this pixel is connected to the initial pixel, and we continue like this until we cannot find any other connected pixel, connected component, which should also have the number 1. And then we start from the beginning, but with a new number. So we find the next white pixel is sitting over there, we mark it with a number 2, and we find all the pixels which are connected to it and label it with the number 2. And then we do the same thing with the pixel intensity number 3. And the very final step is we take all the remaining pixels and put zeros into them in order to have the background intensity 0. And one kind of nice hint, which is sometimes relevant in image processing. If you want to know how many objects are there in this image, it is actually equal to the maximum intensity in this label image. So if you measure the maximum intensity in the image on the right, it's 3, because there are 3 objects in there. Then just a reminder of how this typically looks like. So you start at your initial image, you apply some filters to it, you do some noise removal, background removal. At some point, you do thresholding, and maybe you do some refinement of masks afterwards. So for example, here I'm pretty sure that I applied the watershed algorithm to this data set. And then you go to the particle analyzer or the menu analyze particles, which is doing a couple of things in one shot. It is doing the connected components analysis, and you can output your label map if you select count masks here. But you can also label these objects with our eyes. That is activated with this add to manager checkbox over there. Then you don't get this label map image out, or you don't have to. Then you have for every individual object, individual ROI, you can save individually, analyze individually, which may make, depending on what you want to achieve, things a little bit easier. So there are different ways. Furthermore, the particle analyzer also does some quantitative measurements on these individual labeled objects in your image. So the particle analyzer is actually a couple of things in one conveniently accessible tool. That's why maybe particle analyzer is the most often used analysis tool in ImageJ. Maybe thresholding is applied more often. So very simple tools, very powerful, very often applied. And again, you can get all your ROIs in ROI Manager, and then afterwards from the ROI Manager do some more sophisticated measurements, for example, in order to come to a results table with some quantification from your original data set. When we are talking about quantification, we are talking about feature extraction. So I also showed you some details on feature extraction. I will quickly run through this part as well. So a feature is a countable or measurable property of the image or of the object. And basically there are different categories of features you can extract from your original image. And I'm providing here some examples. So you have intensity-based features like mean intensity, standard deviation of the intensity. You have spatial features and shape features, and you can also differentiate them again, but you don't have to. So think about the area, the volume, elongation, centroid, roundness, aspect ratios. Like we were looking at a couple of things. Then there are mixed features, like the center of mass. I think a student also asked for this one. I see the center of mass as a mixed feature because it takes the intensities into account, while the centroid is just the position from a binary image point of view. So, and that's why you can see the center of mass as a mixed feature. Furthermore, if you take space and time into account, you come to new features like displacement, speed, acceleration, growth, stuff like that. And if you have, for example, several images with different channels showing your cells marked with fluorophores, then we have to think about overlap or relationships between channels, co-localization, stuff like this. Just to take a look at it again, intensity-based features, min, max, median, mean, and so on. We also had this in descriptive statistics a bit more in detail. You can do of images, you can do in regions in images, and if you're still not familiar with the histogram, I would maybe go through an image like that and move a bit a box around while the live button in the histogram window is activated, and look at the histogram and the mean intensity and the min intensity, how it's changing, depending on where you put the ROI and what that means. Furthermore, these position features or geometry features, the center of mass and the centroid and the area and the perimeter and the bounding box, keep in mind that these features exist. They describe you where an object is, how it is internally, that's center of mass, how it is internally weighted towards a certain position, the bounding box, how wide it is, how high the object is. So it's basically also a description of the size and the position in one thing. Shape descriptors, so Farad's diameter tells you where the most distant points of the outlines are and how the distance between these points is. And the minimum caliper or the minimum Farad is like the smallest distance where the object would still fit through in whatever orientation. Then we did not fit ellipses in very detail to objects, but we were using, for example, the aspect ratio, so the kind of an expression of the elongation of an object in order, which is calculated from major and minor axes. And you should, for example, know that the aspect ratio is usually above 1, unless we are talking about a perfect circle, then it's 1. Roundness and circularity are between 0 and 1, depending on if you are looking at a circular round object or not. The solidity is calculated from the object and the convex hull of the original object, and things like that allow you to describe the shape of objects. It's not like that there is this one shape descriptor which is perfect for all the cases. It really depends often on what you want to achieve. That's why it makes a lot of sense to do always this kind of explorative image analysis. You analyze your images, you measure all the shape descriptors, and you figure out if one of the shape descriptors is actually matching to what you are perceiving when you look at the images. And then I would continue using this one in your whole study and trying to report about, for example, shape changes over time. And that brings us to tracking and image registration. So these two terms are here together because they are actually using very similar technologies, similar techniques in order to achieve an image correction or an image interpretation and measurement in images. So tracking terminology, you may remember that we did on the one hand, it's possible to segment objects in different time points and then look between these time points that this object is at the same position like that object in the other time point, or has the same shape, or has the same size, or has the same intensity, right? So this is segmentation and matching objects together. When we are talking about individual points in space and in time, and we want to bring these points together between different time points, we are rather talking about linking these points, even though the process of spot detection and linking is the same, or basically the same procedure as in segmentation and matching. But there's the relationship to image registration, yeah. So we are thinking about different points in space here, not in time, and try to bring that together. And in image registration, we also have the alternative of going via the intensities. You may want to remember that there are fixed images and moving images, and that the translation can be applied to them, and translation and rotation are a rigid transform together. And if you have translation, rotation, scaling, and shearing, this is an affine transform. So the term affine transform refers to the combination of these 4 complicated combinations. And furthermore, if you want to do this locally in different regions of the image, applying different transformations and or measuring these different transformations which have been applied to the image, we are talking about deformable image registration or optical flow measurements or particle image velocimetry. Also a word about machine learning. So you remember this lecture about machine learning? I was introducing you to the fact that we are starting at raw data, we apply a model to it in order to predict a classification. So this is the terminology we use here in the machine learning field. What can be the raw data? The raw data can be pixels. The raw data can be outlines, segmented objects, or kind of pre-segmented things, but can also be whole images of scenes with stuff in there, like a cat and a microscope. Then we are talking what can be the result of a machine learning process. It can be a binarization. So the machine learning experts call this also a dense segmentation. If we're talking about labeling individual objects, so we were talking about connected components analysis already. In machine learning, people call it an instance segmentation. So it's like segmenting different instances of the same thing in these images. We can also classify objects according to their shape, for example. We can differentiate round objects from not round objects by telling a machine learning algorithm to do it for us and by not giving explicit rules as we did earlier. And we can also classify the whole image. So we can, for example, ask in a classification algorithm, is this an image showing a cat or is it a microscope? And the algorithm will do that for us. It's kind of important in a typical use case for machine learning in the microscopy image analysis field is that we have to have some ground truth information in order to guide the algorithm while training in order to do the right thing. So somebody has to sit there and has to outline or segment objects and save them in a certain file format so that we can later on run a computer overnight and figure out the right parameters, the right model, in fact, in order to reproduce this ground truth annotation on different images, for example. And there also the quality measurements play a big role. So this is not a machine learning specific thing. We should measure the quality of all our image segmentation algorithms or results of these algorithms. But here it plays in particular a role because it was not us explicitly defining a workflow and doing a measurement. It was basically the algorithm, the machine learning, the computer who determined a way of finding a good segmentation. And we have to kind of make sure that the algorithm is not misinterpreting parts in the images. And we can do that by quality assurance. Maybe you remember how decision trees work. So basically, that's one very simple machine learning algorithm. So decision tree is basically a combination of these, let's say, nodes, which look at one particular parameter. So we see here in our diagram x2, if it's smaller than 0.3, then the object is a white object. So this is what this threshold here. So this is basically the corresponding number to this. If this condition is true, then the objects are white. So all objects below this threshold are white. And if not, we add another threshold. If it's larger than 0. If it's not smaller than 0.7, so that's above here, then they are also white. And in the final case, we add another threshold on x1, which is in this case here on this axis, and it allows us to build up several decisions, these nodes, into a decision tree, which finally allows us to classify all the objects correctly in this selection, in this sample set of our original set. And if we have many of these trees, making the decision together, for example, in a democratic way, we are talking about a random forest. That's the random forest algorithm. And just that you remember that later on, it's not just one algorithm which is machine learning. There are many different algorithms, and we did not go through them in very detail. I'm just saying, depending on what you want to achieve, there may be a good algorithm for that, or your algorithm may be able to do that, or a random forest may not be able to do that, and then you should look out for potential alternatives. And there's a huge collection, and more of these algorithms appear these days. When it comes to evaluation of machine learning, but also evaluation of workflows in very general, you should always think about the ground truth, the reference object here in this case, which is the green one on the right, and the segmentation, for example, which came out, or the prediction which came out of our algorithm, which is the magenta circle here on the left. We have then these different overlapping regions. So True positive is kind of intuitive there, where the algorithms and the ground truth overlap. This is true positive, so the algorithm annotated it correctly. So they were indeed positive. And here on the right side, the algorithm says negative, but that's not true, that's false, and that's why false, negative. Here on the left side, the algorithm says these are positive, but they are not, so they are false positives. And if you take the number of these events, of these pixels, or of these objects for the classification, if you take these numbers, you can use some math in order to measure, for example, overlap, precision, recall, to figure out, to enumerate, to quantify the quality of this particular segmentation algorithm. And it brings us, the further we go towards quantification, it brings us to biostatistics. So we were going a bit through biostatistics in this semester. And if you remember how is statistics divided, it's basically divided into descriptive and inferential. As long as we just do measurements, so we measure the size of objects, and we measure the mean size of a couple of objects in one image, and we measure the mean size of images in many images, that's all descriptive statistics. We are just doing measurements and we are reporting about them. We do not interpret them yet. We just measure things and tell about them. As soon as we compare different measurements in different images, taking under different conditions, showing different samples, as soon as we start comparing and concluding, so for example, we observe a difference between two different conditions and we conclude that the difference is large enough so that it also must be there in the general population, then we go towards inferential statistics. So we basically, as long as we only report about numbers, descriptive statistics. As soon as we conclude, for example, towards population or towards pharmaceutical working in general, then we go towards inferential statistics. Coming back to our initially shown research cycle, so we start at the research question, we have a hypothesis, we want to design an experiment, then we want to basically execute the experiment, collect some data out of it, have a look at the data or present it to colleagues, for example, then analyze the data in more detail, interpret it, and then having a new question from it. The question we were not able to answer, we have to run another experiment. And here you see again, this is a circle, and it does not mean that we did anything wrong. Actually, when we can complete the circle, we did something right. There are terms like sampling, descriptive statistics, inferential statistics involved in these steps. So just as a recall, then we have different ways of doing descriptive statistics. So for example, we can measure the mode, the median, and the mean of our sample set of our measurements, and they are related to each other. We can also measure the spread. The simplest way was the standard deviation, which is the mean difference from the mean in our sample set. Very simple words. The variance is just the square of it. Minimum and maximum is also easily to determine in case of a practical example of a set of measurements. And between these two, that's the range where our measurements were. And these terms are used to communicate with each other about measurements. That's descriptive statistics, obviously. And we just want to say, yeah, we had measurements between this and that. We had measurements with a mean of this value and a standard deviation of that value. And these terms allow us scientists among each other to communicate in a standardized way how our values were distributed. Furthermore, a student was asking for what the kurtosis means again. Kurtosis describes the shape of our histogram, the shape of our distribution in this particular case. So the mean of a distribution is this particular point. It tells us where it is located. The standard deviation tells us how wide it is. The variance also tells us how wide this distribution is. Skewness, I've also shown in this lecture, shows us if it's tilted to the left and tilted to the right, or tilted to the right. And kurtosis basically shows us, tells us something about the shape of the curve. Is it rather a shape like that, or is it rather a shape like this? And it's not that there is this obvious use case. It's not there's this obvious use case where this can be utilized to measure a particular physiological property of a cell. But if you think about it, the mean the mean of a measurement, it's the so-called first moment. The standard deviation represents the second moment. Skewness is the third moment, and kurtosis is the fourth moment of our measurements. And if mean and standard deviation tell us something, and if the skewness can tell us something about the shape of this distribution, then also the fourth moment and the fifth moment and the sixth moment can tell us something about that, even though it might not be obvious from a practical point of view. But especially when it comes to you have big data, many cells imaged under different conditions, you may not observe a difference in the mean intensity of those cells, but you may observe a difference in the kurtosis of the distribution of the intensities of the cells. And if you see that, so sometimes it really makes sense to have a look at skewness, kurtosis of distributions. And if you see that and if you observe that, then you have to look deeper into why is the third and fourth moment here affected, and the first and second maybe not. So it may, in special cases, may make a lot of sense to analyze this kind of measurements from your original data. Then we were talking about different ways of visualizing data. So we can, looking at tables with numbers makes hardly any sense, especially when it's more than 3 numbers. A scatter plot may make a lot of sense if you, for example, are comparing different measurements, like the box plot and like the Blunt-Altman plot. They show you different perspectives on your data set. If you, for example, see that your data points are on a straight line, so they are more or less on a straight line with some variation, right? That tells you something about the relationship between your two methods. And if this straight line does not fit to the identity, which is this orange line here, then it tells you, if the points are all on one side of this line, then it tells you that the one method is overestimating compared to the other method of measuring something. Yeah? So this is also where the Blunt-Altman plots helps you. So this is basically the same data set shown in a different way. So you see that the difference between these two measurements is in the very most cases above 0. So that means that one algorithm is overestimating the other algorithm. So the Blunt-Altman plot is basically just a different perspective on the scatter plot. Box plots allow you to compare different distributions of different sample sets if they are not pairwise. So if you have kind of independent measurements from this part of the population and independent measurements from another group of another sample. And you should also have from time to time have a look at histograms in order to figure out, at least over the thumb, if this is a normal distribution, because if it's a normal distribution or not plays a role in further concluding from your data sets. Furthermore, Blunt-Altman plots allow us to differentiate different errors we can observe in our data sets. A student was asking for that, I think. What is the random relative error again? So when you look here, it refers to this diagram. When you look at this plot, so basically the values are pretty nicely distributed below and above 0. So it's like obviously these two methods are telling us the same story, but there is a random error between them. So there is some fluctuation. They do not tell us exactly the same value, because otherwise they would sit on a straight line at 0. There is some variation. And this variation depends on the intensity, the value itself. So when we look here from the left to the right, in case we do a measurement of a small thing, of a kid, we're measuring the height of a child, then the variation between these methods is small. If we are measuring something larger, so we are measuring an adult now, the height of an adult, then our error may be larger. And this is what I call a random relative error. A relative because it is related to the measurement itself. When you think about scatter plots, there was also a student asking here, what does it mean if two measurements plotted against each other lie on a straight line? So if they are on a straight line, if they are really on a straight line, it means Pearson correlation will be 1. So this is what this plot here, what this diagram shows us. If they are related, If they are lying on a line, they have a Pearson correlation of 1. That means they are strongly correlated. They can also lie on a line going in a different direction. That means a strong negative linear correlation. And linear correlation basically means you apply a factor to measurement number 1 and you get measurement number 2 out. So this is a linear relationship, like with linear filters introduced earlier. As a simple factor allows you to go from the one to the other. As soon as this factor does not allow it anymore, there is some variation coming from some other source, so it's non-linear, then we come to distributions of points which are not lying on a line anymore, or may have whatever other shape. So this is basically what correlation and linear relationship refer to. Then you may want to remember the confidence interval. So confidence interval is basically something you define. So for example, I want to have my measurements correct up to 95%. Up to 95% of my measurements should be right to whatever definition. And when you look at the normal distribution, for example, here, you can say that the confidence interval of all our measurements lie in this certain range, which is related to the normal distribution. So 95% of our data points in a normal distribution lie between almost -2 sigma and +2 sigma. This is what the normal distribution is actually what it's defined. So in fact, it's not 2 sigma. In fact, it's 1.96 sigma, but maybe we don't have to be too serious about these little numbers. It's just when you look at the distribution of points and you are talking about the confidence interval, this is that means a certain range around the mean where a certain number of data points is in. For example, the 95% confidence interval is the region where 95% of your measurements lie in. And this is a property of the normal distribution, and that's why also in Blunt-Altman plots you find them. Then we were talking about hypothesis testing. I'm testing these days a hypothesis, and my alternate hypothesis is students had significantly different grades when learning online. As they can watch everything on YouTube at any point in time, I assume they learn more. So this is what I would like to prove. This is my hypothesis. That's my starting point for hypothesis testing. And we call this an alternate hypothesis. And the alternate hypothesis is basically the statement which we would like to prove. And then there is an opposite, kind of opposite hypothesis. It's the so-called null hypothesis, which have to be falsified. We have to, from a statistics point of view, we have to show that this one is not true in order to conclude about our alternate hypothesis. So in the case, I would like to show that students had different grades. My null hypothesis would be the students had the same or similar grades when learning via YouTube. And we need kind of this combination of a good alternative hypothesis and null hypothesis in order to do this from a statistics point of view. This also shows again how falsification plays a positive role in science. Yeah, so I will basically, you are about to prove that my null hypothesis is not right. The marks this year will be different from last year. The grades this year will be different from last year. So you are about to prove that my null hypothesis is wrong. And if my null hypothesis is wrong, I can conclude that my alternate hypothesis was right. So let's see how this experiment goes. And there the p-value and also the alpha value play an important role. So for example, before I perform my experiment, I define my alpha, my confidence level. I want to be sure with a remaining error of 5%. So I want to be sure up to 95% that my students really were better this year than compared to last year. And we can measure that with the p-value. So if I reject my null hypothesis because my p-value was 0.01, then this 0.01 expresses my probability, my likelihood of being wrong when doing this conclusion. It is very unlikely that I'm drawing a wrong conclusion, but still possible. So that's what the p-value tells me. And the smaller the p-value is, the more unlikely it is that I'm drawing a wrong conclusion when I reject the null hypothesis and accept the alternate hypothesis. So this is basically what the p-value tells us. So we were talking half the semester about programming. It was my duty to introduce you to programming. And I will not go through all the details again and explain you what all that means, but I would like to recap here. If you are still not confident with the concept of variables, write a simple program like that, execute it, change the values, replace numbers by strings, execute it, and try to get confident with the basic principles of programming, which is basically variables, operators between variables to combine variables, commands like the print command to print something out on screen. You should also, for example, in ImageJ macro, use the auto-completion and read the documentation here of what these commands are doing. For example, if you want to clear the log window here, print clear is your command. Furthermore, macro recording in ImageJ macro is a very powerful technique in order to figure out how a workflow for image processing runs. So don't type everything yourself, record it. It's much easier. And while recording, have a look in the recorder. And after recording, take the recorded program and cut it into pieces where you know exactly what it's doing. So you can here write some comments in between. I'm doing some pre-processing here. I'm blurring the image before I do segmentation. Here I do segmentation, and then afterwards I analyze that. So that's for you. And it's also for yourself in the future. If you are half a year later look at your ImageJ macro, open one of your old ImageJ macros you wrote at the beginning of the semester and figure out if you can still read it. If not, you want to maybe put additional comments in between to write down for yourself what you were doing here, and it also helps others in reading your code later on. So after recording, cleaning up your macro is an important step. Furthermore, I introduced you to the conditional statements, which do something in case a condition is true and do something else in case a condition is not true. And the same thing exists in Python. It just looks a little bit different because of the curly brackets and the colons, which are combined in a different way. If you are still not confident with that, also here open a Jupyter notebook and just try it out with some simple examples to gain trust into this programming language. In Python, the indentation is more important than in ImageJ macro, but in ImageJ macro, I would also strongly recommend to indent your code because it makes it easier for you to read. For loops, if you want to execute the same operation on a couple of images, on a couple of channels, of a couple of objects in an image. These are for loops, and for loops also exist in Python and also exist in Python in a different way. So there is some way of writing a for loop in a more confident to read way. So the usual thing was for all values in a given input set, do this and this and this. But you can also say do this and this and this for all values in an input set. So you can write it in Python and the other way around. Many people love it. Don't repeat yourself. Rather use, for example, an own custom library in Python or an ImageJ macro. You put everything in functions. Things you would like to use more often, you put it in functions. And then you just call these functions instead of copy pasting code and doing the same stuff over and over. Here was a question by a student asking me, if you want to apply numpy.mean, if you have to import numpy. Yes, it's as simple as that. If you want to use something, also on a sheet of paper in the exam, you have to import the right library, otherwise it will not work. So it is as simple as that. And coming back to this slide, I showed you at the beginning of the programming part, lecture 3, I think, and I told you that after some time and some practicing, you will be able to read the program shown here on the right for analyzing the banana dataset. So if I would now remove the comments here, could you still, maybe click on pause now here on YouTube, could you still write the comments there and know what the following code blocks are doing? So I bet you do, because you did this going through a folder of files and segmenting it and doing some measurements and saving the measurements to this. You did this a couple of times during this semester. So maybe at the beginning of the semester you felt unconfident when looking at this thing, and I bet now you can just read it. If not, practice it. take the programs we wrote during the semester, remove all the comments and write the comments again, read your own code and figure out what it's doing, to practice that on a further step. And later on, when you can read code in the same way as you can write code, you will have a much easier time, for example, in your master or bachelor thesis when you want to analyze some data. If you can code this analysis in a Python Jupyter notebook or in an ImageJ macro, some image analysis, you will be very thankful for all the time you can spare, instead of analyzing images manually or doing measurements in an Excel table manually. Code this stuff, and you will be happy. It's also a reminder, keep it short and simple. Some people who start programming become fascinated about it, start writing complicated programs for simple things. You should not. Try to keep programs as simple as possible in order to avoid issues which just come from the complexity. So you see here on the left side and you see on the right side a program which are almost doing the same thing, but they are not, because there is a single typo. Here I can still make this citation. There are two ways for designing software. You can make it so simple that there are obviously no issues, like the one on the right, or you can make it so complicated that there are no obvious issues. So if you see a way for simplifying your 3000 lines ImageJ macro, do it. The shorter a program is, the simpler the commands are, the better it is for you. So whenever you find a way of shortening some code, putting some code together in functions which have a context, do that. Clean up your code, put comments there. That's just hints from somebody who is programming for many, many years. One more word about tools and toolboxes. So I introduced you to a couple of tools, and it's not like that I can show you all these tools in very detail. So one of the students asked what is necessary to know in lecture 5. So lecture 5 was basically only about tools, how every program works. So I introduced you to these tools because you study molecular bioengineering or biology or something related. And I'm pretty confident that earlier or later you will be confronted to microscopes and microscopic images, and you may want to analyze them. But I cannot predict of what kind of images you will be looking at in 5 or 10 years. And I can just show you different tools. So we will quickly go through these tools, and I will tell you in which situation you will use which tool. But at the end, there is no perfect tool, and there is no best tool. And at the end, it only depends which one is the right one and how much you should know about it depends on what you will do next. So, there's no general conclusion for stuff like that. So if you are confronted to automated microscopes which spit out billions of images every day, which all look kind of similar, but all are also a little bit different, then you want to go for Cell Profiler. Cell Profiler allows you to take these billions of images and generate even more measurements out of them, put them into databases and analyze them with modern techniques. So you don't want to program for this large amount of images, which are all a little bit different. You don't want to program individual ImageJ macros for that. You want to have a solid software like Cell Profiler, allowing you to do things like this. If you come to the point where you have to combine different tools, so maybe you have some part of the analysis in ImageJ and you have some part of the analysis in scikit-image, in Python, whatever, then you may want to use a tool which allows you to combine approaches from different other softwares. And KNIME is one strategy for doing that. So KNIME is good if you have from different universes software and you would like to combine it in one common workflow together, then KNIME is a very good strategy in order to do this. If you think about three-dimensional image processing, visualization, and active contours, and it's a very beautiful, and also from a visualization point of view, very beautiful tool to interact with data. Use IC. Also, algorithms can be expressed in this modern way, in this visual programming style. So if you didn't like the coding with lines and comments in between and stuff like that, if you're forgetting semicolons all the time, maybe this way of coding is feels more convenient to you. Furthermore, I showed you a lot in ImageJ and Fiji, so I do basically everything in Fiji. So if I tell you you should use Fiji, then you should have in mind that I am biased because I do everything in Fidschi, but it may not be the right tool in all the cases. But I'm very confident in Fidschi. That's why I can do all the things in Fidschi. It's possible that you, after using KNIME for a year, can do everything in KNIME. So that has really something to do with you, the environment around you, and practice. Become good. So Curtis Rüden said it at some point to me. It was a very important lesson at that point in time for me. Become good in one tool, the one tool which is best suited for your work, or which is very well suited for your work, and be aware of some other tools. You don't have to learn all tools on the same level. You should be very good in one of them, and you should know the others. That in case you run into trouble with your one tool, that you still know the alternatives. And this is basically a very good strategy. And Fidschi is just one out of these many tools. So inside Fidschi, where we're also discussing a number of plugins. So you here see, for example, TrackMate for cell tracking over time. And the student asks, how do I identify well-looking tracks? And how can we judge if a track looks good? If you look at this particular video, so you see here cells dividing over time. They make one division, two divisions, and three divisions. If you look at this video, you see three divisions happening over time. If you then look at the corresponding track, the track scheme window over here, you go from time, so this is time goes down here, and see one division, two divisions, three divisions. These are good looking tracks, and these tracks are looking chaotic, or the divisions happen in time where they do not correspond to what you observed in the video, then it's a not good looking track. So you have to, when using TrackMate, you should ensure that what you see here in this track scheme, that should represent what you perceived in the video. And I think, honestly, it's not necessary to get all these hundreds of cells in here tracked perfectly in order to do a quantitative measurement out of that. If you have something like 10 or 20 good looking tracks, and you can exclude all the other ones where maybe the detection didn't work or something like that, then you can start concluding from this measurement. So you don't have to track every cell perfectly. A small amount of cells is enough in order to continue research and to draw conclusions from that. So it just has to, the track scheme has to represent what you observed in the video. And of course, ideally, you can apply the same strategy to several data sets. Then I introduce you to GPU accelerated image processing. Also here, I'm biased. I programmed that. That's my research. So when I tell you you should use it for everything, then maybe don't trust me. But in case you come to the point where you are working with big data, long time lapses, and it takes years to process that, you may want to consider GPU acceleration because it speeds up things. And speeding up processing means enabling processing. So in some projects could not have been done without graphics cards. So consider this in case you run into trouble like that. Then machine learning, there are different tools for doing machine learning in a image processing context. The most popular is for pixel classification. So if you have an image and you just annotate a bit, this is a cell and this is no cell, you can use the trainable vector segmentation and the LabKit plugins in ImageJ, which allow you to do this. If you want to go further and also say, okay, I have now a good segmentation of my cells, and I now would like to differentiate cells of different shape or cells of different kind with different protein expressions, whatever, then Elastic might be the better choice. And there's online a lot of videos explaining you how to do this. The documentation is really kind of complete. Furthermore, I introduced you to some Python libraries. And there was also a student who asked for all the plotting and table processing and which one is the right library for what. And I'm repeating myself here. I can just introduce you to a couple of tools. I can just show you the door. I can tell you what many people are using. So for example, NumPy is maybe the most popular Python library anyway. So it's like everybody's using NumPy. Many people use Matplotlib for simple plots, but there is also alternatives to that. And again, this comes with experience. So when you did the one or the other project with NumPy and Matplotlib, SciPy, you will gain some confidence. Then you will see that other colleagues or other people on the internet use other libraries. So you can try them out and also get some feeling for them. And so you find your particular way, which is the best for your particular research. There is no such thing as the best library for doing X or the best library for doing Y. My idea was to show you a bit some starting points for different libraries you could use, and letting you do some exercises with them so that you get a bit of feeling what Python in general means. Because Python in general means you import a lot of libraries of different kind and you kind of push data around between these libraries in order to get your analysis done. And if you're looking for a two-sided paired t-test, then you Google for that, you find a library which has it, and you try it out. And if it's too complicated, you don't use it. So this is basically how the freedom of a data analyst looks like in the Python environment. So you choose your tools according to what they do, how well they do it, how accessible they are. And there is unfortunately no such thing as the best tool for doing some particular thing. I also introduced you to image processing in Python. I showed you scikit-image, and I showed you ITK, which is basically an alternative, which comes from the C++ world and from the medical imaging field. If it comes to making a choice between scikit-image and ITK, I would say ITK has, for the medical field, more sophisticated functionality. For example, image registration is very well developed in the inside segmentation and registration toolkit. But it is also more complicated to use than scikit-image because ITK is a proper software developer thing, while scikit-image comes a bit more from the data science point of view, where everything is a bit more accessible in order to get things done right. Then I introduced you to Napari, the three-dimensional image viewer for the Python universe. Maybe at some point it will kind of be an equally well optimized tool like ImageJ. But from the Python universe that the Python developers and the ImageJ developers find together, and can apply all their algorithms in these two universes. So development is going in this direction, but we are not there yet. So Napari is amazing, but there's also some significant stuff still missing. So that's why I introduced you to it. But I also told you that this is a software which is under development, which is an opportunity. You can contribute to development and you can talk to the developers in which direction you would like to go with them, or that you would like them to go with you. So it may make a lot of sense to follow what the Napari developers will do in the coming years. And when we look back, that was a slide at the very beginning. I promised you to introduce you to all of these topics and to all of these softwares. And there's one entry I didn't do. I didn't manage. So I did not introduce you to QPath, but I should have. And that's why I'm doing this now in very short. If you end up in a pathology department, when you look at histological slices, big two-dimensional images, slide scanner data, showing tissues with different stainings, then QPath might be the way to go. So especially big data, very well optimized, machine learning, deep learning, it's all in there. It is a very, compared to all the other softwares I've ever seen, it is a very user-friendly tool where you interact with tools to annotate stuff and then let the computer do the rest of the annotation, doing quantitative measurements out of these annotated images. That's all in QPath. And the documentation online is very complete, and you can also script it, and there is YouTube tutorials. The cool thing about QPath is also that Pete Bankhead, who made it, who is the PI behind this project, can explain very well, and has a cool way of illustrating and visualizing workflows and stuff. So if you are in this field where you work with histological data, so I don't, that's why I may not even be the right teacher for this tool. If you are in such a field and if you are confronted to that kind of data, check out QPath. I think it's much better suited to histology, pathology data than any other tool I showed you during this course. And furthermore, some words about the community efforts behind. So you may have seen already that there is quite some things ongoing in the so-called New Bias Society. So if you find yourself in a couple of years analyzing images every day, you may want to become part of the New Bias community. And also, they have their own YouTube channel. You may have seen that, where we share our knowledge. So that makes a lot of sense. Often, unfortunately, Image analysts are individuals, single people sitting in institutes analyzing images for everyone. And in order to exchange knowledge, we have to have kind of a community. So we meet every year. We have this online platform where we exchange knowledge online. And if you find yourself analyzing images at some point, join the new buyers community. If you're from Europe or not, doesn't even matter, I would say. Just get involved. Furthermore, keep in mind that all the tools I showed you are maintained by the open source community. That's people, scientists, software engineers working in institutes usually, very most of them, doing science on images. And it's people who meet in a room and drink beer together and code stuff. So if you want to become part of this, you're very welcome. Just get in touch and tell us what you would like to contribute or how you think you could help. And so I can just tell that being part of this community is amazing, and sharing workload makes a lot of sense. And I don't have to develop everything myself because other people take care of it, while I'm also contributing my part. So if you feel like becoming an image analyst, if you feel like becoming a software developer of image analysis tools, get involved. And yeah, so furthermore, also a community thing, quite important, just a reminder. Think of colorblind people. They are not so rare. I think it's 15% of the population. So whenever you make a figure, whether it's red and green combined, think about it if there's maybe another way, like combining magenta and green and then visualizing your data like that. And if you do this, you can also not be hit by the red-green microscopy car, which is a Twitter account. I haven't figured out yet who is running it. But if you publish figures which are from a colorblind person, hard to interpret, the red-green microscopy cow may mark this thing. You don't want this. Please make colorblind friendly figures. And also don't forget to cite the tools. So whenever you use an open source tool, whenever you use a certain plugin, please cite the method you are using, the software which allows you doing your science. Please cite it because only in this way developers can continue developing the software because they need citations in order to apply for funding. So please don't forget that. Furthermore, a word about sharing code. So you have developed during this semester a couple of times a workflow where you do some pre-processing, then you do thresholding, and then afterwards you do some connected components analysis and measurements. These workflows have been developed a billion times over the years. And we are developing them again and again. We are reinventing the wheel again and again and again because our community has a bit issues with sharing. So whenever you write a paper, a master thesis, whatever, put your script, which does the analysis, in the attachment. Do not just write about what it's doing. No, put it there, and potentially also put it online, and then just generate. We call it a digital object identifier, a DOI. Generate an identifier for that and cite that. That makes it citable, online, accessible for everyone. And at some point, we don't have to write these stupid little scripts anymore because they are available and we can just download them. So we have to come to a point, at some point, we have to come there where we enter in Google, I would like to analyze this kind of image, and then Google should spit out ImageJ macros which do it so that we don't have to write the same code over and over. But in order to come there, we have to put the code online. We have to share our code. We have to share our experience, especially with the basic stuff, so that we can kind of overcome this and do more sophisticated image analysis in future decades. There's also the forum. Just a reminder, if you struggle later on during your master thesis, whatever, don't send emails to experts because they get a lot of emails, and this communication in private channels is again the same thing as earlier, as I just mentioned on the slide before. Ask your question on a public forum. Even if you think it's a stupid question, just ask there. The people are happy to help, and sometimes very simple things are not obvious in various open source software packages, and we can only change that by talking about it. So what I'm saying is, the more people ask stupid questions, the more likely it is that the software will improve so that these stupid questions will not come up again. But therefore, we have to have these stupid questions, these apparently stupid questions online, because sometimes developers are just not aware that simple things are complicated. So that's why we have to discuss it, and this discussion forum is there for you. So if you want to analyze images and you don't know how, put an image there, ask your question, and you will likely, without any delay, in half a day, you will have an answer from an expert suggesting you a couple of tools you could try, and then you can get started. And it's for free, yeah? So it's like for free. Okay, so what's next? So this course ends here today, so that's the last video. If you want to go further towards microscopy, so that's kind of going in this one direction, there is a couple of YouTube channels I could recommend. So the BioTec Dresden has a couple of videos. And if you end up in a PhD program later here in Dresden on campus, you will end up in this light microscopy course anyway, I would assume. So you can watch some videos from them. Then the micro courses explain a lot of individual parts which are important in microscopy. So if you want to learn what does the point spread function mean and what was resolution again and what is noise in a microscopy context. So that's all there, all in this YouTube channel. There's also the eye biology microscopy course. I think they already do it for decades. Famous people from the microscopy field talking about their microscopy technique or microscopy in general and putting it as YouTube videos online. So you can learn a lot from these YouTube channels. If you want to go in the other direction, doing machine learning and more sophisticated image analysis, there's also a couple of videos I would like to recommend. There's also many of them were also taken as part of the new Bias Academy. Furthermore, especially this join the dots thingy from Dominic White here linked as computer vision. That's really a cool thing because Dominic is basically with a little camera filling out riddles automatically with a Python script. So if you want to go in such a direction, so this is kind of Python programming but fun. Have a look at that. Dominic is a great teacher. Furthermore, more serious image analysis with Python and Jupyter notebooks and scikit-image and Napari. There were also tutorials about all that. Some of them part of the new Bias Academy and some others already longer online. Check them out. If you think about more different ways of image analysis or what you can do with image analysis, or especially how we resolve this reliability issue and how can we trust image analysis in a very general way, I would like to recommend the video here shown on the left from Kota Miura, who is our vice president of the New Bias Society. He has a very cool overview. How should we deal with image analysis in very general in order to be reproducible, quantitative, trustworthy, and so on. So check out the video from Kota. Last but not least, I enjoyed these Zoom meetings actually a lot, especially those with the very live discussions about various tools. And after 14 weeks of sitting in my kitchen and explaining something to my cat and my laptop, I was thinking of how to continue this in a way that we can continue chatting about software. And I was suggesting on Twitter having a podcast. So I open a program and I'm in a Zoom meeting with two other people, and maybe the one has more expertise than me in this tool and the other person has less expertise than me in this tool, and we explore this tool together and talk about what we think, what we see, what we would like to do, what we cannot do, what we can do with this software, and just recording this video and putting it on YouTube. I was just thinking about that loudly on Twitter, and then this happened. So many people volunteered for this. And I would like to thank all the people who volunteered because that makes me quite confident that it will indeed happen. So I think communicating about tools makes a lot of sense. I don't know everything and nobody does. So that's why the only way of how to spread knowledge about using tools in the right way is communicating with each other. And so maybe a podcast is a solution to that. So I think I will start it in September. I have a lot of volunteers to choose from. I think this will be great fun. And one last final word. So some of you will dive deeper into image analysis, maybe become an image analyst in a couple of years and write more sophisticated code and do more sophisticated data analysis. And for those of you who go into this direction, who enjoy doing image analysis, I would like to give you one sentence from Richard Hemming. The purpose of computing is insight, not numbers. Please keep that in mind when you analyze data. And with that, I will close this semester. It was fun working with you, and see you online or see you in real life.